Perfect.
Okay. Good morning, everyone. Thank you for joining my presentation on rapid disease diagnostics in broiler chicken using metabolomics. Today, I'll be sharing how metabolomics can revolutionize the early detection of poultry diseases. This is the outline of my presentation today. First, I will give you a brief introduction to my research. Then I'll talk about my objectives, methodologies, results, and conclusion. As you can see in the left side, infectious disease outbreaks cause significant losses to the broiler industry every year. Imagine as a solution, if we can find a rapid diagnostic test which can detect the diseases even before clinical signs appear, that will be really helpful to prevent outbreaks, reduce therapeutic antibiotic usage, and promote animal welfare and human food safety. And this is where we stand now. The current diagnostic techniques used in the poultry industry has some limitations. If we take serological tests like ELISA, molecular tests like PCR, RT-PCR, and also bacterial culture and MALDI-TOF, none of them can detect diseases as early as 1 to 2 days post-infection and before the onset of clinical signs. But detecting diseases before the onset of clinical signs is very important because time to diagnosis is very critical to prevent outbreaks. So enter metabolomics based early disease detection. Actually, metabolomics is the study of all the chemicals in the body with a molecular weight of less than 1,500 Dalton. And this metabolome is very sensitive to quick changes in the body, and it can detect metabolite changes at cellular levels. And it can explain the metabolic pathways in real time. Most importantly, validated metabolic biomarkers can interpret disease risk, early onset, progression, and also prognosis. If we talk about the current status of these metabolomic studies, in human medicine, such metabolomic studies are very common, ranging from non-infectious diseases like cancer and diabetes to infectious diseases like COVID-19 and tuberculosis. However, in veterinary medicine, the metabolomic-based studies are still in their infancy, including poultry. And most of the studies that are done in poultry based on metabolic biomarkers also were done based on the metabolic disorders of chicken, such as ascites and heat stress. However, the metabolic landscape associated with the viral and bacterial infectious diseases is limited. Therefore, my objective is clear. Therefore, my objective is to diagnose the viral and bacterial infections by identification of metabolic biomarkers as a rapid diagnostic tool. For this, I use two candidate pathogens, one bacterial pathogen E. coli, which causes acute fatal septicemia and high chick mortality, and also the viral pathogen avian reovirus infection, which causes lameness in birds and subsequent production losses. So I carried out separate experiments for E. coli and also avian reovirus. So this is the design for the E. coli challenge experiment. So at day 0, I took 2 groups of birds, control group and a treatment group. And according to this well-established E. coli challenge model in our lab, at day 5, I challenged the birds with E. coli. And according to this model, it usually takes 3 to 5 days post-infection to show signs like pericarditis, perihepatitis, and chick mortality. So I collected the blood from birds before onset of clinical signs. That is, I took the 24-hour post-infection time point. And this is the experimental design for the reovirus challenge experiment. Here again, at day 7, I challenged the birds with avian reovirus using a well-established animal model in our laboratory. And according to this model, it usually takes 5 to 7 days post-infection to show the swelling of the footpad and lameness in birds. Therefore, here I took 2 time points before the onset of clinical signs, that is 24 hours post-infection and 72 hours post-infection. After collecting blood, the serum was separated and subjected to liquid chromatography mass spectrometry. And after collecting spectra, the metabolomic analysis was done using MetaboAnalyst 4 software. And here I must say that we used multiple statistical techniques to obtain the results, like univariate, multivariate, feature selection, and machine learning. And only the metabolites that were found significant across all these techniques were considered as potential biomarkers. And finally, we also did a cake pathway enrichment analysis to identify the metabolite and metabolite gene networks showing potential molecular interactions between these metabolites. And now I will explain my results. So these are the results from the E. coli challenge experiment 24 hours post-infection. In the partial least square discriminant analysis plot, you can see the control group in green color and the E. coli infected group in red color. Here, the infected and the control group are clearly separated in terms of their metabolite features based on the chosen compounds without minimal outliers. And this Venn diagram shows the significant metabolites across all these statistical techniques I used. Here, we were able to identify 19 significant metabolites in the infected group. And during the pathway analysis, out of these 19 significant differential metabolites, we were able to identify these 6 purine derivatives which were downregulated in the E. coli challenged group. Now I'll go to the results of the reovirus challenge experiment. This one is for the 24 hours post-infection. Here in the PLSDA plot, again, you can see that the control and the reovirus infected groups are clearly separated in terms of their metabolite features with minimal outliers. And again, this is the Venn diagram which shows the results across all the statistical analytical techniques. So here we were able to identify 67 significant metabolites in the infected group. And out of these 67 metabolites in the pathway analysis, we were able to find 5 purine derivatives which were downregulated in the reovirus challenge group. These are the results from the reovirus challenge experiment 72 hours post-infection. Here also, you can see that the control and the reovirus infected groups are clearly separated in terms of their metabolites with minimal outliers. And interestingly, in the pathway analysis, we again found that purine metabolism was downregulated in the infected group when compared to the control. So how these metabolites could be downregulated during an early infection, we can explain this using the concept of immunometabolism. Actually, during an early infection, 2 major features of metabolic reprogramming occur. One is for energy production and biosynthesis to ensure faster ATP production, and also biosynthesis for defense response and damage repair. And secondly, the epigenetic changes linked to the immune cell development, activation, and differentiation, which is usually followed by transcriptional changes. And our purine derivatives, which involve in the purine metabolism, also play a crucial role during an early infection. For example, during an early infection, there will be high energy demand and there will be increased ATP production. And also there will be increased DNA synthesis for immune cell proliferation and also to repair the damages caused by the inflammatory responses. Another aspect is the purinergic signaling done by the purinergic receptors. So our purine derivatives of our interest are either essential components of ATP, a critical component of DNA, or they participate in purinergic signaling as adenosine and guanosine receptors. Therefore, the above purine derivatives are highly likely to decrease in the serum during an early infection. Therefore, we concluded that these purines and derivatives are potential or promising biomarkers for early detection of E. coli septicemia and avian reovirus infection in broiler chickens. However, at this point, we found common biomarkers that are shared by the viral disease reovirus and the E. coli infection, which is a bacterial disease. So our next step will be to identify specific biomarkers for E. coli and reovirus infection separately. And our ultimate goal will be a rapid field diagnostic test, which we can take to the poultry barn, obtain some biological fluid from the bird, either blood or some fecal samples, and read any biomarkers, which could help us to identify early onset of a particular disease, so we can take steps to prevent the losses from future outbreaks. And thank you very much. And finally, I would like to acknowledge all my funding sources, my committee members, and also my lab members for their enormous support in my research. And this will be the end of my presentation. Thank you very much. And any questions or suggestions are highly appreciated.
Thank you, Asha. That was excellent. A reminder, you can put questions in the Q&A box. We have 1 minute here. I was just wondering, practically speaking, how would this work? How do you foresee this working on the farm? If this were to all be developed and go through commercially, would the producer be taking a blood sample or would the vet come out? How would you see that happening?
First, before going to the practical stage, actually, we have to go through a lot of steps. Another step after looking at these biomarkers in the lab, we usually do animal trials here. So our next step will be to go to the field, to go to the farms, and maybe with the help of the poultry extension veterinarians and also with the farmers, we thought of getting the blood samples and then validate these biomarkers. And then we can go to the like field level, what happens in the field and what biomarkers, like, is it validated? And then we may be able to put into practice, like, so whenever the farmers report, let's see, in one flock, if they report any abnormal behavior, we can even check the other flocks and see. It's like we could actually use this as screening plus early detection, so that at least we can stop spreading to other farms, and we can do a lot of things if we detect early using the biomarkers.